So the first short talk will be given by Dr. Rona Shaharabani. So Dr. Shaharabani earned her PhD at Tel Aviv University in the field of biophysics. She's currently a postdoctoral fellow at the Hebrew University at the Center for Brain Sciences and working under the supervision of Dr. Aviv Mezar. And so today she will give a talk titled Myelin Packing Markers Through In Vivo Water Gap Mapping.
So thank you for the introduction. Can you see my slides?
Yes.
Okay, good. Thank you again for the invitation to present my work. And today I would like to describe a new model to estimate the myelin water thickness in vivo using MRI. So Carl already explained briefly that the myelin is a multi-layered sheet of lipid-rich membrane bilayers that wrapped around an axon. And I'm going to be as brief and just explain what is important for me. So it is a repeating structure of lipid bilayer stacks with a water layer gap between them. The bilayer lipid membrane is composed of different types of lipids and protein and is about 4.5 to 5 millimeter thick. The water gap between the membrane is about 2.5 to 4 nanometer thick. And we can define the ratio between the myelin water gap to the myelin membrane thickness, which is about 0.6 to 1 in the healthy myelin. Ex vivo and in vitro experiments show that in the healthy state, the myelin has a compact structure with a consistent water gap thickness. It has been suggested that in multiple sclerosis, the membrane undergoes undulations, resulting in larger gaps between the membranes. Now, this feature was overlooked and was never measured before in vivo. Like multiple sclerosis, other neurological disorders can exhibit the same membrane undulation in the myelin. Both small vessel disease, which is highly common in increasing age, and leukodystrophies, a group of rare genetic disorders, exhibit low dense myelin compaction, as can be seen from the TEM images. We ask ourselves, can we use MRI to measure the membrane undulation? Or more specifically, can we measure the ratio between the myelin water gap to the myelin membrane thickness? So we can use the T2 relaxation time measurement to distinguish between several water populations. Usually, we fit two exponents to distinguish between the myelin water, which is the water that is trapped between the membranes, colored here in blue, and everything else, the intra-extracellular water, which is colored in purple. And there are many methods to measure the water pools, and previous studies have shown that the myelin water has a short T2 relaxation time of about 10 to 50 milliseconds, and the intra-extracellular water has a longer T2 time of about 70 to 100 milliseconds. So we developed a new biophysical model to estimate the myelin water thickness from the MRI T2 measurements. Our model is based on two water populations, the myelin water, again in blue here, and intra-extracellular water in purple. We assume that the sum of the two water pools is the total water fraction that we have in the system. We also define a new parameter, the myelin packing ratio parameter, which is defined as the ratio between the water gap, dW, to the membrane thickness, dm. The model is based on two non-exchangeable water pools and can be applied for any multiple echo data. We assume that the myelin water fraction is the non-water fraction multiplied by the myelin packing ratio parameter. Hence, the equation can be described using water fraction and the myelin packing ratio parameter. So we are left with 4 unknown parameters: the water fraction, the myelin packing ratio parameter, and the T2 of the myelin water and intra-extracellular water. To solve this equation, we use several assumptions. First, we estimate the water fraction from additional proton density mapping. Next, we don't solve the model for each voxel, but for a specific region with multiple voxels. Now, in the region, all the voxels may have different water fraction values, but they all have the same parameter values for the myelin water T2, intra-extracellular water T2, and the myelin packing ratio parameter. To verify our model, we designed a new MRI phantom. We manufactured a myelin-like lamella system in which we can control the myelin membrane thickness, dm, and the myelin water gap thickness, dw. We can change the membrane thickness by changing the lipid types we use in the system, and we can control the water layer thickness by using different monovalent salt concentrations. In a simple case of charged membranes, with a separation dW between them, as salt concentration is increased, the membrane, the charges on the membranes are screened, allowing the membrane to condense. And this is a well-known theory called the DLVO theory, which takes into account van der Waals and electrostatic interactions between 2 interacting charged membranes. Since our system is composed of charged membranes, we expect them to behave the same. From the cryo-TEM images, you can see the membranes, the black circles, and the water layers between them, the gray areas between the black circles. And when there is no salt added to the system, there is a large water separation gap between the membranes. When we increase the salt concentration, the membrane condenses. So you can see that the salt concentration increases, the water gap decreases. When implementing our biophysical model on the synthesized samples measured across multiple lipid concentration, each one with different salt concentration affecting the myelin water gap thickness, we find that the myelin packing ratio parameter is fitted with high accuracy to the cryo-TEM measured cryo-TEM measured ratio. Okay, so the model is working on the myelin-like synthesized sample. It's great, but can we do the same thing in the living human brain? We scanned 25 healthy young adults and segmented the brain into different white matter regions. Here you can see 4 of these white matter regions in color. We applied the same model to the living human brain to estimate the myelin packing ratio parameter, the T2 of the myelin water, and the T2 of the intra-extracellular water. And here you see in each column a different white matter region, and the box plots represent the T2 myelin water across these 25 healthy young subjects. We find that the white matter T2 values agree with previously recorded values. Our model fits myelin water T2 to be about 30 milliseconds and about 90 milliseconds for the intra-extracellular water T2. And just to remind you, previous studies showed that the myelin water T2 is about 10 to 50 milliseconds, and the intra-extracellular water is about 70 to 100 milliseconds. And what about the myelin packing ratio parameter? So for our model, the fit of the new myelin packing ratio parameter in different white matter regions is about 0.84, and this value is in agreement with postmortem studies that estimate the ratio to be on average 0.8. So to summarize, we provided a model for detecting myelin water thickness. We modified the 2-pool water model for any multiple echo data and water function measurements. We tested our model on a newly designed myelin-like synthesized samples and in vivo data. The model shows a reliable estimate of the myelin packing ratio parameter, both in the synthesized myelin-like samples, and which was compared to the cryo-TEM measurements, and in vivo data, which was compared to ex vivo measurements. So the proposed model is feasible, consistent, and physically relevant. And we hope to apply and test our model on clinical data such as multiple sclerosis patients, data that we are starting to collect, and on aging brains, data that we already collected. And since I don't have a lot of time, I would like to conclude. Thank Aviv and the rest of the Meta group for their support. Thank you for the opportunity, and thank you for listening.
All right, thank you, Rona. That was a great talk. Chris, any questions?
Yes, we have two so far. One from Jean Chen. What is exactly the clinical importance potentially of this model in multiple sclerosis? So meaning which model parameters have hopefully clinical value in MS?
Oh, yeah. So in the demyelination process, the membrane undergoes undulation. So there is a change between the gaps of the membrane, so the water thickness changes. So if we have an MRI marker that can tell us that we can physically measure the changes in the membrane, we have a marker to showcase the demyelination process. And again, if you have a drug that can resume the membrane to its original compact position, again, we have a measurable method to see these kind of changes.
Awesome. Another question, from Stella Sanchez. Could you repeat, please, what kind of data is necessary for your model?
Yeah, so what I need from the MRI data now is any multiple echo data. So for us, we used a gradient echo spin echo data, so the T2, and also gradient echo data, the T2 star, and both can be applied for the model. Also, in order to assess the myelin ratio packing parameter, we need the water fraction maps, so PD maps.
Awesome. I'll have one more question. You showed through your sort of phantom studies that ionic concentrations varied the lamellar packing of the myelin. How do you expect this to sort of hold at the postmortem data? So we know a lot of our metrics change when we look from in vivo to postmortem. So you're using that as sort of a reference guide that your metrics were valid. Do you expect change there, or is there enough protein structure that you expect that to sort of be maintained ex vivo?
Okay, so actually I'm just using the ionic strength or the changes in the salt concentration just to make sure I change it to the water layer thickness between the membranes. I'm not actually monitoring, I am monitoring the salt concentration that I am adding, but it's not the same concentration levels measured ex vivo. I can use other techniques to condense the membrane. I choose the salt concentration just because it's a convenient method to condense the membrane. It's not actually a measure of the ionic strength within the system or the ex vivo compared to the ex vivo.
Okay. Maybe one more question from Mustafa Buhara. How do you get your PD map? And can you simply use the multi-echo data to estimate the total water content?
Wait, can you say that again?
Can you use your multi-echo data to estimate the total water content that you use in your model?
No. So the multiple echo is one sequence requirement that we need for the model. the other requirement sequence is the water fraction. So it's the same MTV that Aviv mentioned before. It's a different set of measurements, not related to the echo data that we acquire. And if you would like, you can contact me in email, and I can send you the papers. Aviv already mentioned them about the MTV or water fraction mapping.
Excellent, thank you. Maybe in the interest of time, we'll move on to our next speaker.
Yes,
Sure, thank you.
will do. Thank you so much, Rona.